Cuando quieras, Josemi.
¿Cierro aquí o...?
Vale. Good morning and welcome to this seminar. We thank Marcos Pita, who is the speaker today. We know each other for a long time because we studied the degree together. So before starting, just I will say a few words about his career. He did his PhD at the Institute of Catalysis at CSIC from 2002 to 2006 on nanoparticle engineering for the biomolecule immobilization. At that time, he developed a biosensor able to detect nucleic acids specific for VI8 and swine flu against single mismatch sequences, which was patented and licensed to the company Solutex. In 2007, he joined Clarkson University as a postdoctoral research assistant, and in 2009, he went back to the Institute of Catalysis with a Ramón y Cajal contract. After that, he achieved his tenure in 2013 and incorporated in January 2015 with a permanent position at the Institute of Catalysis. And in that time, he incorporated photocatalysis to the bioelectrochemical processes to facilitate water electrolysis of hydrogen photoproduction, photoelectroproduction of oxygen, and more recently, carbon dioxide reduction and phosphorylation reactions linked to the ATP production. He has supervised 3 PhD students, and he's supervising 3 more at the moment, and he teaches in the Master of Applied Chemistry in the Autónoma University of Madrid. He has published 100 scientific articles and 11 book chapters, collecting around 5,000 citations with an H index of 44, and he has co-authored 4 patents. Today he is going to speak about bioelectrocatalytic systems for water splitting, hydrogen revalorization, and carbon dioxide reduction. So, Marcos, whenever you want.
Okay, thank you very much, Esther. Good afternoon, everyone. Thank you much for coming, and also thank you for joining online to this seminar. I come from the Catalysis Institute, which was formerly a department in this Physical Chemistry Institute, long time ago, even before I started my career. And now it has a separate institute, and somehow it's very warm welcome for me to come to this kind of mother house. And well, as Esther said, this is our group, Bioelectrocatalysis Group. It was founded in 1987 by Professor Victor Fernandez, and he retired in 2015, just when Professor Antonio López de la Cey and myself took over. And since then we have been working in several issues related always with using enzymes, which are biological catalysts, as together with electrochemical systems for different applications. Currently we have a Marie Curie postdoctoral from Iran in our group, and also a postdoctoral former Marie Curie, now in other kind of contracts, and several under PhD candidates. Some of them are about to finish this year or next year, and others are starting. And also we have the pleasure to get a new entitled scientist joining this year early. So we're a pretty crowded group for what we used to be. And our research lines always focus on, not this one here, on the characterization of these redox metalloenzymes. We focus on working on enzymes that have a metal site and are going to make oxidation and reduction reactions, because there are other enzymes that catalyze different kind of reactions like hydrolysation or esterification, etc. But we work only with redox metalloenzymes. And we focus a lot in the interface of these enzymes with different kind of materials, especially electrodes, either gold or graphite, And also we are starting to use semiconductors for photo-derived applications, which is much more challenging. And we have also worked a lot with nanostructured kind of materials, like carbon nanotubes, graphene derivatives, or different kind of nanoparticles. And our ultimate goal is just to generate clean energy, which is not producing high amount because these materials are what they are, and they have their scalability has not been tested yet. But we can use them for different applications related with the high added value synthesis, and by using bioelectrosynthesis, or get compounds that are very expensive to get by common chemistry. As I said, I normally we started this line focusing on energy systems. This is a picture I took myself from my house several years ago. It's in the outer of Madrid, Colmenar Viejo. You can see how looks at 7 a.m. Sky is clean and everything is nice, beautiful city of Madrid there. But from my working place, situation changed in the middle of the day, and this is where we are living. You see that when it doesn't rain, pollution is a major concern that we should have. Okay, this is a motivation for us to try to get different ways of getting energy. And one of the pollutants we have here, everybody knows, at least everybody at these levels, I suppose we know the challenge that is the greenhouse effect caused by carbon dioxide. Even so, we can see that in the last 50, 70 years, carbon dioxide concentration in the atmosphere has gone up. It's very alarming, and in 2021 or 2010, we are always saying that we have to accomplish how to tackle this problem. And we have the last agreement before all this pandemic and all these war issues we're living now is to reduce, like, let's say, at least 50% of the emissions, and to get by 2050 the values we had 10 years ago. However, we're still using 80% of our energy demand comes from fossil fuels. So we need to develop different technologies, not only the renewable energies we can find in the market, for reducing this emission and its concentration in the atmosphere. Here is another graph. Data come from European Union on how the energies are used nowadays. We can see that in the last 20 years, 30 years, we are still depending a lot on oil and natural gas. Renewable sources are growing a little bit, but it's not enough. We need to find different kind of energy supply. So any kind of technology, not only the most promising, but also the most alternative and bizarre ones that we can say use biological entities for developing energy systems, are welcome. And please help with this, okay? So this is where we should move to a carbon dioxide-free energy market. So coming from this appealing to what we want to do, or the final goal, what do I work? And I'm going to present some results related with biofuel cells, captured fuel cells, way to develop energy, using enzymes as a catalyst. Also, we'll present some works on photobioelectrochemistry, and finally, the synthesis of high added values and some ways to reduce carbon dioxide into more interesting compounds. In the first set of work to present, I'm going to focus on the oxygen reduction part of biofuel cells using an enzyme called laccase. Laccase is an enzyme it can be found in trees or in fungal. Fungal are much more interesting for these applications. Normally they are used industrially, so they are very interesting and important enzymes in industry. They normally are used in paper industry for whitening and treatment of paper, because normally this enzyme, its function in nature is to degrade lignin, which is a very hard compound polymer from trees, and it's very difficult to treat and to get value from lignin, but it's always there. So when you're going to use materials from wood or from plants, you always have to face with lignin, and nobody knows really what to do with it. But these enzymes are coming from different kind of fungi that eat these things. So we take the enzymes coming from there. There are other enzymes, there is a consortium, but for our applications we work with this. Why? Because we're interested in the other reaction. How does it degrade this polyphenol polymer is going to reduce oxygen to water and use it as an electron acceptor by using these coppers that are here divided in two active sites. And this reaction, oxygen reduction to water, is very unusual in nature. Most enzymes that are going to use oxygen as electron acceptor are going to produce hydrogen peroxide. This is not the case of this enzyme. And there are very few enzymes that do this reaction. So we say, why don't we try to immobilize it on an electrode and try to communicate these electrons here to get use of this as an high redox potential cathode. Okay, so we need to do what is called direct electron transfer, that the electrons are going to jump from here to here directly. It's very beautiful, but when we face normal in reality is this, that enzymes are absorbing on electrodes by any other kind of orientation that is not what we want. So this is partly most of my job consists on convincing these enzymes to do whatever I want, but normally they do whatever they want. It takes months or years to do these things. Okay, so what can we do to make the enzyme mobilize the way we want, to make this communication? We have to analyze the surface of the enzyme. Then normally the surface of the enzyme, what it does is to make the changes in the active site to achieve the potential and to make it work properly. And it has been a matter of evolution for millions of years to get this optimization. And we want to work with it out of its environment, so it's going to be a little bit more difficult. So we have to analyze to see what can we do. And we find in this particular enzyme some carboxylic residues that are in this site here that are going to, if we immobilize to the electrode, we will do like this. And also some sugar residues that can be used with different kind of immobilization strategies for doing this. But of course we have a challenge here, because in the other side that are going to be driving to this disposition, we have a lot of lysine residues and more carboxylic groups, so the chemistry to make the oriented immobilization is not easy at all. But we could do it, and it was a work for several years to achieve this immobilization. Okay, and we have a lot of work with gold that is very challenging, but I'm not going to present this here today. I'm going to work how to immobilize this enzyme on graphite electrodes, which is much more easy. Why? Because I said in the beginning, this enzyme, its natural reaction is degradation of polyphenols. Okay, it takes the polyphenols of lignin and make the oxidation of it. In the surface of a graphite electrode, we have a lot of aromatic rings and aromatic derivatives that are suitable and resemble the phenol rings that are present in lignin. So the enzyme is going to naturally orient and put the interesting active site, the T1 site, on the surface, facing it. Okay? And later, when it is there, we can modify the surface. In this case, we are going to make a reaction here that is generate this in situ, this derivative with the diazonium salt is going to make a radical reaction here, and later we reduce the nitro group here to amino and make a shift base. Okay, not shift base. This is an amide bond. Okay, this amide bond is going to be from one of the external groups of the laccase and covalently immobilized, because if we don't immobilize it, it's going to be floating in the solution, and the electrochemical system is not going to work. Okay, so when we do this, this is the reaction we get. We are not feeding this with lignin or any kind of phenols, but when the enzyme is immobilizing the surface, what we are going to get is this kind of potential at this 600 millivolts plus 200 from the reference electrode is like 800 millivolts. We're going to get a cathode that is going to reduce oxygen at such a high potential. This is very unusual, and normally in fuel cells with different kind of catalysis, this process starts at this potential. So those batteries are going to have a more difficult, less potential, applied potential. In this system, we can see that making this easy reaction, we achieve like 200 microamperes per square centimeter of reaction. But what happens if we put a mediator? This is a molecule, a mediator that is going to transport and shuttle the electrons from the enzyme to the surface. In this case, we can see that it's going to show the enzymes that are not properly oriented, because we can try to convince the enzyme to be in a good way, but it will always be some portion of enzymes that are not oriented well. And so here we can see this is direct electron transfer, we get this increase in the negative current, and when we put the mediator, we can see that more or less half of the enzymes are not properly oriented. Okay, you can see this work was from 2008, so we can tell you that in the next 5 to 10 years we have been improving this. But this was, for me, the beginning of this very interesting work. How to improve it? Well, we can use nanostructured electrodes and use strategies of immobilization that takes 2 steps. First, to make an orientation affinity, and later catch it with a covalent bonding. So how are these things with nanostructured electrodes? We used, in this case, carbon nanotubes that are hierarchically nanostructured. It means that they're fibers that are micro size, and nanotubes are grown on the surface of these fibers, getting this kind of hairy electrode, very conductive with very big area, and make exactly the same kind of reactions. Okay, so in this case we made an amide bond forming this, and this is what we get. The currents are increased from 200 that we had before or to 500-600 here. Here we realize that there is an issue with the oxygen supply because the enzymes are eating it, and oxygen is not very soluble in these solutions. So more or less this is what we can get. You can see in air, when we put extra oxygen, the currents are going up. And when we make a 2-step immobilization, in this case what we do, instead of making directly an amide bond, we're going to make a partial oxidation of the sugars in the surface of the enzyme to open it and make aldehydes. So then it's going to join the amino groups forming a Schiff base, and then it is linked because the most of the sugar residues are in the proper situation, orientation, favoring that a covalent bonding later. We can see that the increase of the current is like 1.5 milliamperes, which is like more than 5-6 times the values in the work before. Another line of work is to modify the graphite electrodes with gold nanoparticles. We have been using, in this case, 2 different sets of gold nanoparticles, different sizes, one that are on average 16 nanometers and others that are in 5. The strategy to immobilize the nanoparticles is similar to what we use with enzymes. We make a modification here to make a covalent bond, gold-carbon bond here. So then the gold is fixed because it can give problems if it's just absorbed on the surface. And later we make an organic layer here for a scaffold and orient the enzymes. This we learn from different the gold strategies immobilization for immobilizing this enzyme here. So we can see here, this is a part of the electrode that we strip away from the surface, and what is the difference when we have the covalent bonding and the absorbed nanoparticles. We can see that the population of gold is higher, and also after making these activation cycles of gold, we can see here this is a carbon electrode modified with gold. The typical signal of gold appears on the carbon electrode, and the longer we put the nanoparticles on the surface, the more concentration of gold we get. So this peak here, that is gold oxide reduction, is increasing. And there is also another advantage. We could see that when the particles are just deposited by absorption, after making these cycles here, they aggregate to each other. So it's not nanoparticles anymore. But when they are covalently linked, they remain independent, mostly. Because gold has a very high ductility and mobility, and it is a little bit tricky. But it is much better result for here. And what response we had here, it was really surprising for us that the electrochemical response, we compared at different times the 3 hours deposition of gold time. When we put oxygen, we were really surprised here that appears this peak here, because after 10 years working with this enzyme, I have never seen it before. And what can be this? So we have to study this sharp initial thing and when it appears. And we found out that it appears when a lot of nanoparticles are put there, because they depend on deposition time. We can see this process also when oxygen is supplied as an extra, not just in equilibrium with air. So we also compare the nanoparticle size with the smaller ones with 5 nanometers are giving a much better current than the ones that are 16 nanometers. Okay, so this is because the size of the enzymes, that is mostly the same as these small nanoparticles that are 5 cubic nanometers. They have a much bigger concavity. The curvature is bigger, so 16 nanometers starts to be similar to a flat electrode from the point of view of a small enzyme. For us, it's a nanoparticle, but for enzymes, they start not to be so much like curvature, so they start to behave similar to what is a gold electrode. But we have this process here that is giving this process here. We analyzed that this was a process that could be separated in two different kind of processes. One that depends on the tilting and orientation, that is the one that is giving the second one here. But in this first jump here, the electron transfer is not limited by the orientation, but it is really wired, and the electrons can jump directly from the electrode to the active site of the enzyme. So the kinetic limitation is not this step that is normally the one that is more difficult. So as I said in the beginning, using these systems for large energy applications is today very far away from the real world, because there are systems that work in much bigger scale. And it doesn't mean that it cannot be reached, but we have to find applications for our kind of energy production. So this is why we have been working in several European projects for making implantable autonomous devices that are requiring energy, taking energy from the fluid, from the living entities, like not only humans, but not only mammals, but also from plants. So you can take even from juice and other kind of systems. But we were working like, can we make this enzyme that, as it comes from a fungi, its optimal pH for working is acidic, very acidic. At a neutral pH, it doesn't work at all. So in collaboration with the group of Miguel Alcalde, that is devoted to directed evolution, they were developing an enzyme that shifted its pH working range from to more neutral values. Okay, so we could see that this enzyme at different kind of biological fluids like human blood or plasma, or as Corby acid was keeping pretty much of its activity. This one is like a buffer, mimicking buffer with a mediator. So the activity is not zero, it's larger than zero. So this is what we were looking for, and in this way it was a serendipity moment, because we found this, like we were working at neutral pH, because of course not in a, we're going to make physiological conditions, so then we change the working conditions. How does it work? We were looking for this process here, the reduction, but we found that that never appeared ever before. And they said, hey, what is this oxidative current? Because these enzymes are not going to oxidize anything. And at this potential, we have tried many lackases before, and we never got this kind of signal. So this opened a new research line for me that is related to water oxidation, because we wanted to see, like, hey, what is it possible that this enzyme can oxidize water to, or hydroxyl anions, to make even hydrogen peroxide or oxygen? So we wanted to see, because we didn't find any other explanation for that signal there. Water splitting is a very well-known reaction since several decades ago, and well, this is Physical Chemistry Institute. Everybody knows water splits into hydrogen and oxygen when energy is application. This is the theoretical minimum, but as an endothermic reaction, it goes out to 1.5 electron volts. We have to apply to separate water and protons into hydrogen, which is a very good fuel candidate. We are interested in energy fuels that are not depending on fossil fuels, and oxygen, well, is a good gas for breathing. It's always wanted. Okay, so the bottleneck of this reaction nowadays is the much more difficult part. The oxidation of production of oxygen, the oxidation of hydroxyl, requires 4 electrons involved in the mechanism, while protons reduction is a much more easy mechanism. So we thought we had a shot to make a new improvement in this part of this reaction. In nature, we only find this enzyme here, this photosystem 2, that is able to produce oxygen. It has a lot of subunits, and this is the cluster of the active site of this photosystem too, and it's very big. It's unstable. It only lasts like half an hour, and it's very difficult to immobilize on electrodes. And searching in the literature, we found that there are copper complexes described that can be used for water oxidation. This is, well, Texas nomenclature here. And they could use it, applying a large potential for these complexes. And we say we have an enzyme that has copper inside. Its active site is copper, so maybe it works. There is some kind of intuition here, and we say, okay, let's see what we find. And we mobilize the enzyme here at neutral pH and apply very positive potentials. We can see here how did it work. This is the enzyme we have been using the whole time. The initial reaction, this is the oxygen reduction reaction that we use as a tester to see how the enzyme is performing. And then we put into the oxidation cycle, we see that we have this big current here. And later, the life of our enzyme has decreased half, so we're affecting our enzyme with these large potentials. But after 18 hours, we could still get back some of the activity. We also tested how it's going to react if we put a natural inhibitor. That is, this fluoride is going to sit in the path of the electronic transfer inside the enzyme between one of the two active sites, but it didn't affect at all. It was giving the same response. So we assume that only one of the active sites was involved in this reaction, and this is the production. After half an hour, we can see that the currents are higher, and we have to find a way to measure if these currents are resembling oxygen or not. Again, we can see here that after this chronoamperometry, this is the response, the natural response of the enzyme before and after, so the activity was going down. So this enzyme we are naturally used because it's the best one for oxygen reduction, was not very good for water oxidation. It was inactivated and the reversibility was not very high, and then we decided to go again to our collaborator and ask for a mutant enzyme. Maybe if it is has a higher resistance, we can use it for, it was going to perform better, and it was like that. This 2B enzyme was performing much better than trimethylhirsuta hirsuta laccase. The other one was at these current values at the maximum, so it was more than double. And after 30 hours, it was still more active. And here we can see in an experiment in function of time, the higher current we were getting here. And I have to develop a method to measure the enzyme, and I make this enzymatic way to determine both hydrogen peroxide and water here by making this ABTS reaction with making this enzymatic cascade. Okay, so we could see that the oxygen that was measured and translated as this colorant, this a dye, ABTS, it was more or less the same hydrogen peroxide production for the enzyme with the electrode with no enzyme and the electrodes with different enzymes. But the Coulomb efficiency for the mutant one was a little bit higher. Anyway, it's not a very big, 10% of Coulombic efficiency is not very optimistic value, but it's good enough to prove that the system, it can be done. And of course, there is a lot of things to research here for different kind of enzymes containing copper that can make this reaction here. So we say maybe this is also due to the high potential we have to apply. Is there any way to reduce this amount of potential, this high potential we have to apply? And we start to think of mixing the system with semiconductors that are going to take this energy from light. So then we have to apply less energy, and this not so hard for the enzymes and not so damaging. So we start to be in this research field that is related with the splitting of water and the use of semiconductors. The typical example, we never use it, is like titanium oxide. This reaction was published like 50 years ago, and we didn't like it very much because of two reasons. You need to implement ultraviolet radiation, and ultraviolet is very bad for biological entities, even catalysts. This is why we focus on sulfides that are going to absorb visible radiation, which has not only lesser energy to damage the enzymes, but it has enough energy for when we compare, in this case, indium sulfide. We can see here is the conductive band, the ground band, and this is in a very good place for us. It's an N-type semiconductor, and also is going to absorb these photons that reaching the surface of Earth are much more abundant than ultraviolet ones. So we were very optimistic about this, synthesize these particles with very porous ones. The high porosity, we can see 40 square meters per gram, and this is the shape they have, between 20 and 100 to 100 nanometers. And here is the calculation of the band gap energy. This is slightly above 2.1 electron volts. So we mobilize this between the electrode that is expressed here. We put a layer of this material and later immobilize the enzyme on the surface. And now at this time we improve our system. No more enzymes for checking how much oxygen we're producing, but we got an external sensor that is going to tell us how much are we going to produce here. And also very important, how much can we decrease these 1.2 volts we have to apply for making the system work. So here we get some answers here. First, the enzyme, this laccase needs to be covalently immobilized. When it's not immobilized, here the currents are very low. We can see that it's comparable. The electrode is fluorine doped tinoxide modified with the semiconductor. Current increase is very low. And when we put just physically absorb the enzyme, it's also very low. But when we make a covalent immobilization, we can see that we have this increase in the surface when we put the light here. Here it is under dark conditions, this blue line, and here illumination. Interestingly, in this experiment that takes 50 seconds to make this cyclic voltammetry, oxygen is not measured until we have a delay of 50 seconds. We assume this is because it has to go from the electrode surface to the sensor. So did we manage to decrease the voltage? We can see here that when we apply an external voltage of 1 volt and we go down 0.187, the oxygen detected in the sensor was detected until 0.8. Here the potential was not high enough to supply the electrons that are going from the semiconductor to the enzyme. So then, as there is no replenishment, the reaction is stopped. So we can see here at these high potential ones, we still get the oxygen signal in our external sensor. So we managed to decrease from 1.2 to 0.8, 400 millivolts decrease in the system. So, okay, let's try a different kind of sulfide here. So we did the same for tin sulfide. Tin sulfide has this ground level, that is the one we're interested in here, a little bit more deep than the one from indium sulfide. It's also an N-type semiconductor that is good for these applications. Okay, so exactly the same system, but in this case we also implement here indium tinoxide nanoparticles, because we were scared that these particles, that they are like 100 to 100 nanometers, the enzyme are much smaller, they are going to sit and have not very good connection with the electrode, so we have to fill it with something. So in this case we tried ITO nanoparticles that are 30 nanometers to fill in the gaps between the semiconductor and the electrode. This is how the particles look. You see, like 500 nanometers, they have this hexagonal shape. And when we put the ITO nanoparticles, we can see that we go from 0.01% increasing in 1 order of magnitude until 10%. This is very big, so capacitive current here is going to mask everything. So we decide to stay in 1% concentration when depositing the particles, which made a good increase. So this is the response we got here. This is the illumination signal. We get a photoresponse from the electrode, photocurrents, and we can see here, this is from dark to light, we can get better currents here in the cyclic voltammetry, and this is what our sensor is showing us. It was very, very little amount of oxygen. And also, if we could see that if we decrease the intensity of our lamps, this 81.4 corresponds to 1 sun in our latitude. Okay, this was much too powerful because it was not decreasing the intensity. We could see that the signal was even bigger from the base level to the upper level, that if we have bigger, higher, more energetic irradiation. So, okay, maybe we're putting too much light there. So we started to decrease the potential to see what happens there, and we have and also the light intensity. Now at this one, we could see that if we decrease the potential, here the oxygen signal we're getting in our sensor was going up. And we could see that the Faraday efficiency and the oxygen produced was bigger. So we found out that not only applying a very high potential is not more efficient, it's also damaging the enzyme and is making secondary processes that we couldn't, we didn't have any way to determine what was it, probably photooxidation of the material, but it was working much better with lesser external energy application to the semiconductor in terms of electric potential. Okay, so we have been talking a lot about oxygen production, so we also work on hydrogen production, the fancy part of this. We use an enzyme called the Hydrogenase. We have two sources. Here normally we use now this one, the sulfur ones with selenium, and we want to connect it to different kind of semiconductors. First of all, I working with photosystem 1, not 2, that is the one that produces oxygen, but photosystem 1 is present in plants and is responsible for absorbing light in chloroplasm. So we developed this system here with an electrode modified with an osmium polymer that is going to transfer the electrons to the photosystem 1 that are pumped with the light here, and is to a secondary polymer that goes to the enzyme, and the enzyme is producing hydrogen. We tested here two polymers, one with methyl biology derivative here, and another one with cobaltocene. Here are the redox potentials. We can see this is the osmium polymer, this one here, and these two. We were comparing which one is going to connect better with hydrogenase. And of course, we found that the cobaltocene one was responding much better. Here we could see that when we put the light, we get the production of hydrogen. Normally, when there is no light, it's at minus 0.4 volts, and we're producing them with light. In this case, very small. But here in this case, even at the potential of the photosystem 1, we start to see the production of hydrogen here, which is resembled by these reductive currents. We have very high hydrogen yield, like 83 picomoles per second of hydrogen. And we also tested the enzyme with our semiconductors, also not only with photosystem 1, but this indium sulfide, we could immobilize the hydrogenase on it, and in solution, in the absence of any kind of electrode, we could get response using a sacrificial electron donor. In this case is sulfide. When we put the enzyme here, we can see that we switch on the light, and then this is hydrogen production measured in real time with a mass spectrometry. When we switch off the light, the system stops, and we switch on, and we goes up. And here is an analysis of the hydrogen production measured by mass spectrometry after its incubation time, putting together the hydrogenase and this indium sulfide particles, reaching the maximum after 6 hours. And also it's very interesting that the photoactivity corresponds to 90% of the photoproduction of the hydrogen produced here. So it's a more efficient time, incubation time, the optimized time here. But we had a problem because as indium sulfide is an N-type semiconductor, when we put this on an electrode, it didn't work because the electron flux, the excited electron flux, was going the other way. This is why it was with laccase for oxidation, but not for hydrogen production. So we were working with this like 1 year and no results at all. And we had to find, decide, okay, we need a material that is also in this kind of band gap, that the conduction band energy level has to be above, more energetic than the couple between hydrogen and protons if we want to produce hydrogen. And very important, it has to be a p-type semiconductor. Okay? So we found a ternary sulfide here, that is this copper gallium sulfide, that is, it has all these requirements here. So we characterize it by XRD to find how to immobilize it on the surface of the electrode. This is the way it looks under transmission electron microscopy. And it also can work in solution. We can get the same result here. We can see that when we put the hydrogenase in the surface of this copper ternary sulfide, it can take the electrons from sulfate here, pump it in, and produce hydrogen. And produce with the mass spectrometer, we could see also this hydrogen production. It is lower than the one with indium sulfide, but also the irradiation was lower, because in the other one was an early experiment. We had the first experiments we did with these illumination reactions. That lamp was working at 450 watts, which is a lot, and now we're working at 80, and of course the production was lower, but the trend is the same. We can see that the production of hydrogen serves the same way, but it can be mobilized on electrodes. Okay, so we test different kind of immobilization techniques. I'm going to go a little bit fast through this because most results are really hard. We mobilize how to modify the electrode here. First, by drop casting, we can get, after different cycles of drop casting, how it is going to put the ternary sulfide here. We mobilize the enzyme just by letting it to absorb, which for hydrogenase works much better than for laccase. And this is the kind of response we were getting here. Okay, so when we make the illumination, we get this tiny photoresponse, which is to us, okay, not very high. So what if we do a spin coating? Pues more or less we're getting the same kind of response, a little bit higher, but still not good enough for us. And later we try to put carbon nanotubes on the surface of this. We put here, modify to see if we mobilize the enzyme on the carbon nanotube, it gives a better response, and we have to make the illumination. These electrodes are transparent. We only cover them through this side. We mobilize this way here, and we get this kind of response here. Also, in some cases, it's not very good. Okay, and we found that if we put a mediator, we could see the response of the mediator. So we found, okay, this is very difficult to immobilize this material on the surface of the electrode. So what is happening here is that the response is independent from the hydrogen production. Almost, we have an overlapping of hydrogenase immobilized on the semiconductor, which are only a few and produce a little bit of hydrogen, and other ones that are directly immobilized through the gaps in the surface of the electrode directly. So when we go to minus 0.6, this is the natural potential, that is not light assisted, we get this response. And when we had the light assisted measuring at these potentials here, the response was very little. So we were very angry with this. More than 2 years. So another synthetic method, how to can we assure the coverage of the electrodes? So we make the synthesis of this ternary sulfide in presence, putting in the reaction chamber the electrodes to modify them. And still we get different kind of response. You can see the trend is changing here when the potential is only applied for measuring only the photoresponse, and not here we have the mix of both photoresponse and electrical response directly with no photo assistance. So what else can we do, because this was not leading anywhere? So in the end we found work for making electrodeposition of this. So we just put the precursors here and making cyclings of electrodeposition at different potentials, which is this process here. During 15 minutes the electrode was now giving a much better response. So this is ongoing research. I still don't have all the data to show, but we can see that we went from 1 microampere or less than 1 to 13 microamperes. So we are now working on this direction. So we can compare here how much is the hydrogen produced in all of these systems here. So here is nanomoles, the rest are picomoles after 1 minute of application of project. So I'm sorry, I still have to finish this research line, which is promising but hard. So what can we do with the hydrogen we produce? Because normally in industry they think with hydrogen has very large amount for making something, right? So we're going to make other kind of transformation in value in products, adenosine triphosphate, or make phosphorylation reactions that are very interesting in research. How are we going to do that? We have a collaborator that supplies us with this enzyme here that is ATP synthase. It lives in the membrane of the mitochondria, and it has a head here that it turns. So it takes the adenosine diphosphate, cutting off this one, and reversibly can produce the ATP or make the opposite reaction. How does it do it? It depends on the pH gradient we have at both sides of the mitochondria. So normally here the pH is more acidic, and there is a proton pump that is going to make this phosphorylation. So can we cheat this enzyme and say, hey, you are not on the mitochondrial, you are on an electrode, and we produce protons here on this side. This is what we want to do. So if we produce protons in this side here, we're going to cheat you, and you will understand that here is a lot, and you will produce ATP. And for doing that, we're going to mix this with hydrogenase, and a special one that has a lipidic tail. This modification here will allow the enzyme to put the lipids here in this side and making these electron shuttles here that are normally used for pumping electrons to the active site that is here. So this was the perfect enzyme for us to put inside the system. As I said, here is the electron path to the active site and the lipid anchor here. Well, this is the hydrogen production active site here. And also this enzyme is unlike other hydrogenases tolerant to oxygen, partially tolerant, let's say. So let's characterize this. We put the enzyme here. How does it work? As I said, here is the electron path, the active site, and the lipid bilayer covering the electrode. Can we put protons here and make these reactions in this side? So here you have atomic force microscopy. You can see the lipid bilayer on it, have this cover, and if we remove it with a detergent here, then we can see these protuberances here that correspond to hydrogenase. So we were pretty sure that this work was more or less okay. So if this is the electrochemical response. When we give hydrogen to the system, we pump hydrogen inside, we can see the oxidation of hydrogen. But if we put nitrogen, there is no electrochemical response because the enzyme is not oxidizing the hydrogen and giving us current. We also wanted to see if we're going to need a proton gradient, we need a way to look, because it's very easy to measure the bulk pH with a pH meter, but how to measure the local pH under a membrane? So we developed here a monolayer of these compounds that they can dimerize after application of a slide potential. Here we're going to produce this is going to be a redox probe. It can oxidize and reduce this amino to imino at here, and depending on the pH here, this is going to shift a little bit, like this. We can see that when we put this monolayer, this is going to move with the pH towards here. We can see this is the potential sweep, and it is a linear relationship here. We can measure this, the local pH under the membrane. Control experiments showing that if we have only the membrane, we put hydrogen there, there is no way to oxidize this hydrogen, so the peak doesn't move. But if we put the enzyme and we don't cover it with this bilayer, if we put hydrogen, it's going to produce, but protons are going to the solution. So then there is no shifting. But if we cover it with the lipid bilayer, then we're going to make a higher proton concentration here, and we can get this one. We can this shift here. And when we put an ionophore, that is a molecule that is going to break this, is going back. So we had a way to calibrate the local pH and the bulk pH. Later we studied how to work with this ATP synthase and how to perform the systems. This is the quartz crystal microbalance characterization. This corresponds to the deposition between liposomes with no enzymes and liposomes with the ATP synthase. We get this amount, this value is the mass corresponding to the ATP synthase. And what can we measure how to improve, how is a pH change here? So if we give ATP, we want to do the other thing, but for characterization purposes, we supply ATP here, and it's going to dephosphorylate it and pump protons inside. And we can see that after 2 hours, there is a potential shift in our electron probe in the surface of the electrode. So in this chamber here, in this interstitial space, is going to make an acidification. The whole system is going to work like this. we're going to supply hydrogen. Hydrogen is going to produce protons here, and this proton is going to force the ATP synthase to produce ATP. Again, you have here this characterization by atomic force microscopy. This is a bare gold electrode, the hydrogenase electrode, and also we have the ATP synthase. And this is the response we have, the electrochemical response. We can measure the hydrogen oxidation. It's going to give us this oxidative current here. We get a limit because of the diffusion of hydrogen. And this is related to the ATP production we can measure here. Okay, so we can measure the ATP just by supplying with hydrogen. So the whole system is moving this. This ATP, we link it with these two different reactions. One for phosphorylating glucose. Glucose phosphorylated in the market is like 3 or 5 times the price of this. It's normally used in different kind of research systems, but it's a good model here. So we could see that there is which one is we wanted to know which one is the limiting step here in this. The production of ATP is the slowest reaction because increasing the amount of ATP supplied to the initial regeneration system is going to be here. This one is going to give back adenosine diphosphate, which is going to be taken by the ATP synthase again to produce the ATP, and we can phosphorylate glucose, and it goes much faster when you have a lot of ATP in solution, so the catalytic amount of ATP is a little bit high. And we could see this reaction here later by oxidation and putting this compound here is going to be absorbing light at 340 nanometers, so we can monitor this reaction spectrophotometrically. So we supply with the hydrogen, and we are producing the ATP. The ATP is taken by hexokinase, and glucose 6-phosphate produced by hexokinase is taken by this glucose 6-phosphate dehydrogenase and producing this. And we can see here the production when all the system is, but as a control experiment, if one of the enzymes is missing, there is no glucose 6-phosphate produced. And more interestingly, we can see that after 2 hours, if we supply with 500 micromolar ATP, here we have produced 600 micromolar of glucose 6-phosphate, which means that all ATP molecules have at least once, and some of them twice, participated in this catalytic regenerative system, showing that the system can work. More interestingly, is using a different enzyme, not only for glucose, but for NAD phosphorylation, because this is extremely expensive component used in biotechnological industry. So in this case, we use the same. We produce the ATP the same way, and with this enzyme here, NAD kinase, we can produce NADP. Later, we take a different glucose 6-phosphate dehydrogenase, okay, that is going to use it. Only it doesn't use NADH, but only NADPH. So this one is not interfering here. We took us several months to find this, and then we could see how it is working and producing NADP, and we could see again that if we supply only ADP, the reaction is producing NADP this way. And if we put an initial amount of ATP, of course, the reaction is faster. So we can transfer hydrogen into a very high value product with a lot of interest for pharmaceutical industry. I don't know how I'm in time. This is the last part of my talk, so I have, okay. So you want me to stop, or it's Okay, so the last enzyme we have been working here is formate dehydrogenase. That is an enzyme that normally takes formic acid to produce carbon dioxide at the end of the respiratory chain, but it can be used reversibly. Which advantages it has? Oh, sorry. That it has a lot of selectivity. It only produces formate. That is a single product is one of the major challenges for carbon dioxide valorization. So this enzyme is very similar to hydrogenase. Okay, so it has a path and goes to a tungsten cluster that is going to reversibly perform this reaction. This is mediated electron transfer work we publish on gas diffusion electrode. But the work we have really been doing in our lab, not in collaboration, it was for direct electron transfer. We decided to go to graphite electrodes because in these reactions, gold is not very good because normally it's modified with thiols and they are desorbed, so we cannot use them. But we can modify with diazonium salt and immobilize the enzyme here. And we can see here the response we're going to get for oxidation of formate. So we can see that with a mediator we have high current, but we have direct electron transfer here. Also, we can oxidize, and it can work also in a reversible way. This is the reduction of carbon dioxide to formate that we could do in different configurations. This is with a mediator, and in direct electron transfer we could produce formic acid with this enzyme here. And here we just make a measurement in time to determine by integration how much is producing here, and we determine that almost 100% of Faraday efficiency is going to formic acid here. Cross-linking is not improving the system because of the enzymes are somehow affected, and the best way is just to immobilize directly here, and we're working on this. So this is the end of my talk. These are the guys that are paying this research. Okay, coming from national funding, regional funding, European funding. A lot of collaborators for doing these things because we don't produce the enzymes, normally they produce. And most important part here, if you like bioelectrochemistry, in 2 years we're organizing in Alcalá de Henares a World Symposium about this. So hopefully, if you like and you get into this field, you can see it. So thank you very much for your attention, and now I'm open for questions.
Thank you, Marcos. We have time for questions or comments or any doubts you can have.
Sorry. Thank you very much, Marcos, for this for your talk. I think that one of the main concerns when dealing with immobilization of enzymes or proteins in support in general is how to get an oriented immobilization. And I don't know if did you try to use some tags in the protein to either in the N-terminal end or C-terminal end of the protein to immobilize. For instance, the typical one is a his tag. Did you try the use of tags for oriented immobilization of, for instance, laccase?
Yes. Yeah, sometimes we have, for laccase is not so easy. But for other enzymes like peroxidase or hydrogenase, they are modified with groups for histidine tagging, for streptavidin tagging, but it's also a challenge because it sometimes works very well, but normally, well, you need to make this enzyme, then it's not from a wild organism. If you want to put a his tag, you have to make it in a bacteria culture or yeast, and we normally don't have access to that. But we have worked with some of them at some point, and especially for orientation characterization.
Yeah, but what do you mean? That the proteins you are using are not recombinant proteins, are commercial ones? Or
some of them come from direct evolution, so they are mutants, not even recombinant, but mutants. But some of them, the hydrogenase is natural, and formate dehydrogenase is also a natural one. And the laccase, it's also from fungal natural ones. We have been testing, because we have another research line with a collaboration with these groups for characterizing mutants they do, and they normally supply with, normally the most common modification we use is to add some cysteine for immobilizing on gold directly, because the cysteine is going to expose a thiol, and you need to put a monolayer. And you still need to put a monolayer, because if not, the enzyme is going to attach, especially on gold. It has multi-phase sorption, so the enzyme unfolds. Because a lot of groups are attaching, and then the tertiary structure, quaternary structures are unfolded, and the enzyme loses a lot of activity. So it's not so straightforward. The idea is is very common, but later the results are not always
Yeah,
as good as one would like.
I was somehow indirectly related with some immobilization of enzymes, and I would like to that you comment some aspect which always I was very interested, is that in many cases when you try to a covalent immobilization of an enzyme to support, how do you deal with the fact that some enzymes, through its catalytic mechanism, needs some conformational changes? In many cases, multipunctual covalent immobilizations somehow, I think, may restrict these changes. So how do you deal with this with aspect?
You are completely correct, and we try to avoid multipunctual immobilization. We normally use single point immobilization through a covalent bond, and we still lose activity. Probably the enzyme is slowed down because of these steric restrictions. We also, especially with these systems related with semiconductors, not only with electrodes, some of the enzymes, especially hydrogenase, works better if we don't immobilize it, because the material is extremely porous. So we guess that, I don't like to say guess, but it's very difficult to see how an enzyme is immobilized inside a material. But there are a lot of pores, so the enzyme gets there, and somehow it finds that if the electron doesn't go here, goes to the other side of the semiconductor, it should work something like that. But in the end, the activity that it shows with the semiconductor is like 15–20% compared to its natural activity inside a cell. But inside a cell is useless for us. So so we have to deal with that. But still it's pretty good activity.
Okay, thank you very much.
Thanks to you.
Marcos, how do you prepare the sulfide nanoparticles? It's chemical synthesis, and do you have a control on the size distribution, or does it have any effect in any case?
Yeah. The most important factor here is not the size distribution of the particles, but the pore distribution. That's the most important part, and that is why the synthetic methods are solvothermal methods, and it has been a lot of optimization, research. We just come at the end of that. So our collaborators in the photocatalysis department here have been working with this, and we just say, hey, you prepare this, let's use this, and we learn how to do the successful way. And we skip all the hard part. But they were trying like a lot of surfactants, like Triton or other kind of polymers. And later purification is really hard because it's difficult to get out of the surface these polymers. But okay, you can do it, and well, that's PhDs of collaborators of us, not ours. But
And the gold nanoparticles are commercial, or are synthesized by you or your colleagues or anything?
We made them.
How?
In the case I presented here, there are two synthesis methods, one with citric classic reaction, and the other one we use a specific steric hindering reductant that is a phosphoderivative, has a boronyl with 3 phenyl rings that is going to make more difficult to the gold to grow too much. And also we know how to synthesize them inside of inverse micelles. I don't know if you are familiar with this, but this is an organic solution with tiny drops that are controlled by the size of a micelle. We can make them from 1.9 to 4 nanometers, and they are extremely monodispersed. But you have to later, I mean, this is very beautiful work. That the gold is formed there, is stable inside, and later what we do is to add a thiol molecule that attaches to the surface of gold that has a carboxylic group or an amino group, and we modify the pH. So it is deprotonated in the case of carboxylic groups, or protonated in the case of amino. So this becomes very unstable inside the organic solution. So you can make very easy a liquid extraction.
Okay. More questions? There is a comment here by Miguel Ángel Treviño. He says that when you are desalinating seawater, a problem is that you obtain acidic byproducts. A mixed factory for desalination and hydrogen production by hydrogenase could be an added value for both processes. It's not a question, but I don't know if you have any comment on this.
Seawater.
Yeah,
Okay.
he says that a mixed or a combination factory for desalination and hydrogen production by hydrogenase could be an added value for both processes. I mean, I think
Yeah,
he's related.
it could be, but you have a problem in seawater because there is a lot of oxygen, and these enzymes inactivate with oxygen. So, I'm sorry. I wouldn't put too much effort on that because the enzyme is not going to be active unless you can cut off the surface of the electrode, the oxygen, which is possible. There are works from our competitors that they use polymers for covering these hydrogenase electrodes with a specific polymer that works in two ways, as a mediator and also as an oxygen scavenger. So the enzymes don't get inactivated. I don't know how stable those polymers would be in seawater, but I would start that way.
Okay. More questions or comments? No? Okay, then we thank Marcos again for coming and giving us this seminar, and we have a small present for you.
Thank you. I'm flattered. Super mag. Uy, sí.